My name's Anna. I'm a master's student at Purdue University working with Thomas Hook and Paris Collinsworth. And today I'd just like to kind of share with you guys what I've been finding as a part of my master's research. And that research involves investigating lake-wide distribution, diet, and growth rates of larval alewife in Lake Michigan. So for those of you who don't know or are not familiar with the alewife in Lake Michigan, the alewife are an open water schooling species originally from the Atlantic Ocean. Um, they have migrated into the Great Lakes through the St. Lawrence River, through Canada, and then through the Welland Canal, which is kind of famously known for the Niagara Falls area. However, by the 1950s, alewives became increasingly abundant in Lake Michigan, overwhelming the Lake Michigan food web. I've circled them here on the food web just to kind of get you guys oriented with this diagram as it comes back up a couple times. These alewife, however, did not do so great for Lake Michigan when they first got here. They outcompeted native species like bloater, which is on that same row that they're on in the diagram. And eventually the numbers rose so high that there were lake-wide die-offs, which some of you might remember. That one of them happened in the 1960s, and then we had another one in the 2000s as well. So in 1965, the fisheries managers initiated a salmon stocking. Specifically, Chinook, coho, and rainbow trout were introduced to compensate for the loss of those native top predators like lake trout. The goal of this was by introducing predatory fish, managers aimed to reduce the overabundant alewife population through biological reproduction. And the outcome was successful. The alewife population declined as those top predators reduced the pressure that those alewife were having on the lower Lake Michigan food web. Um, thus meaning that those once considered invasive threats, alewife are now considered critical non-prey species, and those support Lake Michigan's vast recreational salmon industry, which I'm sure a lot of you guys are interested in. While that's all happening, there's been a pretty important impact to the lower half of the food web, shown here in that red rectangle. So phosphorus is a limiting nutrient for photosynthesis. It fuels phytoplankton growth, and those phytoplankton are the base of the aquatic food web. So all those larval fish are eating these phytoplankton to grow energy to get bigger. Historically, in Lake Michigan, phosphorus loading came from things like detergents and untreated sewage, and fertilizers, and they helped produce these things called algal blooms, which some of you might notice in the not just Lake Michigan, but the other Great Lakes as well. Since the 1980s, though, phosphorus levels have declined in Lake Michigan, largely due to regulations on point source pollution, so kind of nipping it in the bud where the root of pollution comes from, and as well as the Clean Water Act and the Great Lakes Water Quality Agreement. So basically, this reduction in photosynthesis inevitably means less food for the phytoplankton, which also means less food available for those higher trophic levels, or those, in other words, those alewife, those yellow perch, and eventually those salmon and trout and whatnot. While this is all happening, I'm sure a lot of you have been hearing or have heard about the mussel problem or the descended mussel problems. So introduced in the 1970s or 1990s, apologies, these mussels are pretty impressive filter feeders. They remove phosphorus, plankton, and bacteria. So not only are we regulating phosphorus input into the Great Lakes, but also these mussels are slowly filtering out all the important nutrients for those lower level trophic levels to be feeding on. And with no known natural predators, they've dramatically altered the energy flow in the lakes, impacting everything from zooplankton or those lower level food web animals all the way up to the Chinook salmon, resulting in lower food availability for bottom dwelling invertebrates, which can affect sport fish that rely on them. So what? Why should you care about this? This is all to say that the Lake Michigan has gone through some pretty impressive changes. So not only are the phosphorus levels lowering, that again affects the bottom level of the food web, those phytoplankton. There's also invasive mussels, those quaggas and zebra mussels, who are constantly filtering out plankton and nutrients from the water, making the water look clear, which aesthetically is phenomenal, but for at a biological or ecological level is pretty impactful on the energy flow that happens in Lake Michigan. Um, it's also the food web's collapse has had the collapse. So the alewife introduction of alewife um, has a pretty impressive impact on the way that energy is transferred through Lake Michigan. And alewife have outcompeted some other important fish in the Great Lakes. This is all to say that that all includes has a bigger impact on the sport fish. So specifically, salmon and lake trout have less to eat, changing in both all sizes of fish, numbers, and where they're caught. And for the anglers, this could be fewer fish, smaller fish, changing hotspots, which means it's harder to fish. So understanding the ecosystem on my end helps protect the future catch on your end. This is all to say that all these things have a great impact on fish. So specifically, what I'm interested in is the early life history or the larval stage of these fish. During the early life history or the early life stage of these fish, they are growing the fastest. On this diagram, it's a quick little diagram I put together of total length on the y-axis and years in ages on the x-axis. I've circled that first year 0 and I'm zoomed in on that because actually during the first years, even the first few days, the fish is growing the fastest it's going to grow its entire life. That's a good thing because faster growth means higher survival. Fish that grow quickly in their early lives are more likely to survive, that meaning larger fish are better equipped to avoid predators and find food. This early life, if the fish aren't growing, that's going to be an impact on the recruitment or the addition to the stock. So poorer growth means lower recruitment, means again that those fish are not being inducted into the stock, meaning that declining growth rates leads to a reduction in population numbers. For alewife, historically, there have been really low growth rates on the millimeter per day scale observed in specifically the southern Lake Michigan. Again, that slower growth means higher vulnerability to mortality. During those first few stages, if they're not growing fast enough, they're susceptible to pretty much everything. Predation, um, uh, upwellings in the water, current weathers, like light penetration has all an effect on the mortality and the survival of these alewife. For you, again, thinking about that bigger picture, that Lake Michigan food web system, low growth and survival rates of alewife suggest recruitment issues. So if those alewife aren't recruiting to the stock, your salmon and your lake trout and your other trouts don't have the ability to feed, and then those fish don't have the ability to grow bigger for you guys to catch and eat yourselves. With fewer surviving to adulthood, we can expect to see again a decrease in population of your favorite fish. We can't move forward. We can't begin to discuss the future without looking kind of at what's been historically published in the literature. To orient you to this table on the left, you have year. So your left. So year in ascending order, and then location, which I really wanted you guys to pay attention to. All these papers have been researching the southern half of Lake Michigan, and while this is great, we don't really have a good understanding of what's happening in the north. And I also wanted you to pay attention to these bottom two rows right here. These are the results from the paper that was most recently published on alewife growth, diet, and whatnot in Lake Michigan. Again, southwest, southeast, which is great to know. Doesn't really tell us the whole lake-wide picture. And that growth rate is sitting right around 0.48-ish millimeters per day. If you look up on that table, these fish were once growing at 1 millimeter per day. So that drop from 1 millimeter to 4.4 millimeters is pretty significant. And part of my research is to figure out where we stand more currently. So how are we going to study this alewife recruitment issue in Lake Michigan? Me specifically, we decided to collect larval alewife in Lake Michigan. Using those alewife, we're going to estimate age and growth. And then we will also conduct a diet analysis to kind of understand better what's happening around the fish. What are they eating? How are they eating it? And to what degree are they eating those little zooplankton? Again, just to kind of recap everything that I've just said, Lake Michigan has gone through some really impressive changes. Those invasive mussels, the the phosphorus input regulations is increasing that water clarity, pushing all that nutrients, uh, clearing up that center of Lake Michigan, meaning the nutrients near shore has become slightly more abundant. That, coupled with what we're seeing in that decrease or that 0.48 millimeter per day growth rate, is leading us to try to characterize distribution, diet, and growth rates of larval alewife collected through different regions and bathymetric depths in Lake Michigan during 2021. So to do that, unfortunately, I didn't have the pleasure of collecting any of these fish, although I would have liked to. The Cooperative Science and Monitoring Initiative, or the CSMI, which is a binational effort aimed at coordinating science and monitoring activities in one of the Great Lakes every year, meaning that once every 5 years, Lake Michigan gets sampled for whatever the managers in that area have decided that needs to be checked up on. So in 2021, Lake Michigan was up, and they decided to collect um, samples from 15 different transects that's located here on this map. Each of those little dots represents one distance from shore. So at each of those transects, they sampled 7 distances from shore. And at each of those distances from shore, 3 layers in the water column were sampled. For the purposes of my study and for my brain, I broke up Lake Michigan into these quadrants, which you can see here on the color coding legend on the side. Those quadrants are important and those carry out throughout the rest of the presentation as well as the rest of my research. It is important, however, here to note that the lake-wide survey was conducted between July 4th and July 26th, 2021. That'll be important in a second. At each of those distances from shore and each of those depths, larval fish and zooplankton samples were collected using the 2 nets that you see here on your screen. However, I wish I could have been part of this. The larval alewife were then identified, enumerated, and measured by members of both the EPA and the USGS. And once that was done, all those fish came to me. And the best way for us to be able to understand age and growth rates are by using otoliths. So for those of you who aren't aware of what otoliths are ear stones in their fish's ear. They are made of calcium carbonate and they lay down just like the trees on a ring. And that's exactly how we count them. They're used for a couple different things, but most importantly, they're used for aging. Again, we count those rings like we would on a tree. However, the big difference between the otoliths that we are using in my study and that you would see other people are using in their studies is that ours are for larval fish. So each of those rings that you see on that otolith on your screen represents one day the fish has been alive. Whereas in an older fish, each ring equals 1 year. So we're working with a time scale that's much more refined than what we would be using if we were using an adult fish's otolith. By counting those rings, we're able to get an age, and with that age, we are able to use an equation to determine our growth rate or how fast the fish is growing per year. In our case, per day. You can also use otoliths to understand environmental indicators. Otoliths can be broken down and used for isotope analysis, which I personally, have not used, but that's a pretty important aspect of otolith research. Again, for the purposes of my study, we're using age and growth rates to better understand how these fish are moving and growing. So for each of these fish, again, you can see the fish on the top of the screen here. That's about how big they are. That's about how big the fish that I'm working with are. We decided to pull the otoliths from larvae that were bigger than 6 millimeters because underneath 6 millimeters, they were just too small and we weren't able to see anything. For the clarity, for my study specifically, we decided to take multiple pictures of the same otoliths, stack them on top of each other in Adobe to create one clean image in which we were able to use to count those rings. Growth rings were counted by 2 readers. Actually, Dom and I were the ones who did these. And growth was estimated. Age and length were used to estimate growth. Total number of 2 was added to all the ring counts as alewife do not lay rings their first 2 days of life. Going into some results, I just first want to talk about distribution or densities of alewife in Lake Michigan. Again, important to note that these were collected during July 4th to the 26th of 2021. This was all these samples are considered a snapshot, so this is not a very long this sampling event did not resample every day. They took one sample and then moved on to the next transect. First, right out the gate, again, I mentioned we did not have any historical data on the northern part of Lake Michigan in regards to alewife. So this is pretty cool to see that we're seeing higher densities both near shore and in the northern region. Northern region. Specifically, we're seeing large numbers in Waukegan, Sturgeon Bay, Manistee, and Grand Traverse Bay. There are a few hypotheses that my advisors and I are kind of running through to try to figure out why those areas are specifically higher than others. One running theory that we have right now is that a study performed by a Purdue University student found that the spawning time of the alewife was in late June, specifically the southeast region. So that we think we're not seeing a lot of fish in those areas because we missed that huge spawning event. And by the time that the boats were out there sampling, all the fish had kind of gone offshore to grow up and live happy lives somewhere else. Moving into our growth rates, so we used a pretty simple equation to determine our growth rates. So we took the length of the fish, we subtracted 3.5 because that is how big the fish alewives are when they're hatched. And we divided it by the age. So what you see here is length in millimeters on the y-axis and age in days on the x-axis. From a total of 400 fish, we were able to calculate a lake-wide, so again, this is not broken up into any quadrants or anything, an entire lake-wide growth rate of 0.72 millimeters per day. Don't worry, I'll make sure that we put that into context in a little bit here. Another theory that we kind of wanted to see was whether distance from shore had any effect on how fast these fish were growing. So to orient you to this graph a little bit, on the y-axis or that tall axis, we're going to have growth rates. That's our millimeters per day. And then we have quadrants on the x-axis. And each of those little graphs, those 5, 8, 12, 17, 23, 30, and 38 boxes, those represent the 7 distances from shore that were sampled through this project. As you can see, because all of those boxes within those distances from shore boxes are kind of all along the same level around 0.72, we didn't find any significant difference between the fish that were caught offshore and the fish that were caught near shore. This is leading us to believe that there was really no difference, that distance from shore played no role in how fast these fish were growing, despite the offshore regions being significantly clearer from nutrients and zooplankton than the nearshore areas. Moving into our diet analysis, for diet, we took our same fish that we conducted otolith analysis on, and we extracted their guts from their bodies. And working in sections, gut items were ID'd, photographed, and measured. Here are 2 images that we collected from a fish that we sampled here at Purdue University, just to give you an idea of what we were looking for inside these bodies. Just quickly, presence, absence. So alewife don't consume a lot in their first couple of days. So we weren't really expecting a ton of individuals to have gut items in them. We actually found that 30% of our total 524 had something substantial in their stomachs. The rest 70 were empty, and this makes total sense based off of how small these fish were. This is also pretty on brand of what we were expecting, with Eberheimer et al. Being the study that was most closely related to the work that I'm doing, in which they found 33% of their 313 total fish to have gut items, and 67% to be totally empty. Oop. So just kind of running through the conclusions of what we found so far as part of my study. The distribution of these alewife, we're seeing pretty high numbers in the north. That could be due to a number of factors. We're still trying to kind of figure out the biology and the ecology behind those. We also think that we missed a potential larval emergence, or that larval spawning event in the southeastern quadrant, leading us to have significantly lower numbers of alewife in those specific areas. Here is that graph again, and I've added my study on the bottom of it, showing that yes, while in this 2019 study, we see that there's some pretty low growth rates in larval alewife, that lake-wide, our alewife are growing a lot faster and historically it's more accurate to what we were seeing in the past. However, in all the things that we were looking at, distance from shore played no role in that growth rate. So no matter where these fish were caught, near shore, offshore, they were growing about the same rate. And for our diet, we compared to our past studies, our alewife are consuming about the same amount as they were back in 2015 when that previous study samples were collected. And what does this mean for you? Again, trying to make this relevant for you by understanding alewife growth and habitat use, or the prey item that salmon are going after. It helps us to predict the future, guiding stocking efforts and boosting your chance of reeling in that trophy catch. And with that, I'd like to thank all the funding and collaborating partnering agencies that helped me with this research, as well as the members of the Hook Lab for their assistance, as well as the undergraduate technicians who have spent hours and hours and hours staring at those little alewife.